Hi girls, this is a review of Mendelian genetics. So I'm just going to do this by going through a practice problem. Okay, and this will help you with that practice problem packet if you're having any trouble. So we're going to do a problem with guinea pigs. So in guinea pigs, black hair is dominant over white. Cross a homozygous black guinea pig and a white one. Okay, what would be the phenotypic ratio of the offspring? And if two of the F1s were crossed, what would be the phenotypic ratio of the F2? So let's go through and first talk about dominance. Okay, so we need to understand, in order to answer this question, the law of dominance. Which basically means if you have two or more alleles, the dominant allele is going to be expressed. Okay, so in this question, I have black hair and I have white hair, and black hair is going to be dominant over white. Okay, and we represent dominant alleles with capital letters. So a recessive allele is represented with a lowercase letter. Okay, so for this example, black is dominant, white is recessive. So it's really important to kind of make a key. Keys really help you for solving problems. I know we're only working with one gene here, but making a key can be really helpful. So remember that all organisms are going to have two copies of each gene. Okay, and these copies can be the same allele or they can be different. If they're the same, we say homozygous. If they are different, we say heterozygous. So for our example, we're working with a homozygous black guinea pig. So same allele, the dominant allele, so we're going to have two copies. Okay, you could also have homozygous for our recessive allele because we're not expressing any of the dominant. Okay, we know that the white one, little b, is also homozygous, but this one is going to express white because it lacks a dominant allele. The other condition is heterozygous, which we may need later, but this is going to have one dominant allele, one recessive allele, and this one will also express black, okay, because one dominant allele is present. But they are different alleles, so we can refer to it as heterozygous. So this is a little bit of the background information you need in order to solve these problems. And remember, we're going to be referencing meiosis a little bit today. It's really helpful to kind of help us solve some of these genetics problems. Okay, so let's start writing out this problem. So we have a homozygous black guinea pig, and we're going to cross it with a homozygous white guinea pig. Okay, we know that the white one has to be homozygous because it's not expressing a dominant allele. I'm just going to label this one as the female and this one as the male. It doesn't really specify in the question, but just for purposes of talking about meiosis, it's going to be helpful. So in order to solve a genetics problem, the first thing we need to figure out are the gametes that are going to be produced. And remember, okay, based on the law of segregation, okay, each parent can give one allele, not both. Oops, where's the screen? And this is the result of the separating of homologous pairs. Okay, and this happens during metaphase and then anaphase 1. We're separating homologous chromosomes. That's the whole purpose. So this parent right here, I know they're identical, but we're just going to play along. Can give this bee in an egg, or this bee in an egg. So half there would be this parent. Can give this, or this. Okay, this would be a little sperm. Okay, and just to kind of bring us back for 1 second, so we remember, okay, so this parent, so if we just focus on the mom here, remember she has 2 copies. Okay, they're going to be identical because they're homologous, but she has 2 copies. They replicate during the beginning, the S phase. So we still have 2 B's. Okay, during metaphase 1, they line up. Anaphase 1, they're going to separate. Which will give us, I'm just going to show this one side. It's a little bit easier. So the resulting cell would have 1. Okay, we separated our homologous chromosomes. During anaphase 2, these little guys are going to separate, resulting in 2 of these. Actually, technically, I have 4 of these. Okay, so I'm going to produce 4 of the same gamete. Okay, they're going to be exactly the same, but I could do this twice and go the other direction as well. But remember, the reason we do this is because of meiosis, and we're segregating gametes. I know it's kind of weird to do it whenever it's the same gamete, but it'll make more sense and be helpful a little bit later. Okay. I need to put this little second question down. And plan my space well. Okay, so here is our problem. And we'll come back to law of segregation in a second, and it'll kind of make a little bit more sense whenever we're working with something heterozygous. So we want to demonstrate the possibilities for this organism. Okay, so there are 2 possibilities for eggs. Okay. I can give little b or big B. So all of the offspring are going to get, okay, you kind of just bring your alleles in. You guys have probably done this before, but just get in the habit. Okay, and all of my offspring are going to have big B, little b. They are all going to be black. And if I wrote my phenotypic ratio, okay, this is my F1 generation. If I wrote my phenotypic ratio, I would have 1 black to 0 white. Okay, basically all black, 100%. If I wanted to go through and do the next generation, okay, my F2, the F2 is, you're able to get it through crossing 2 of the F1s. So say I have 4 offspring, maybe 2 males, 2 females. Okay, they're all going to be big B, little b. If I cross 2 of those together, I can figure out what the offspring are going to be for my F2 generation. Okay, remember each parent can give one, not both, so they can give a big B or a little b. Okay, so this is the female, this is the male. And you can have a big B or a little b in the egg. You can have a big B or a little b in the sperm. Okay, and these would be the gametes. You want to use those gametes to make your Punnett squares. Okay, each one of these little boxes is going to represent a possibility for the offspring. Okay, these would be the eggs. These would be your sperm. Okay, bring this egg and sperm together to make a new individual. So making zygotes here. Okay, so if I'm looking at this, you want to think phenotypically. That's the most important thing, is the phenotype. The genotype is important, and it'll be more important when we get into other types of inheritance, but you want to think about what you can see. So phenotypically, okay, anything that has a big B is going to express black. Okay, the absence of a big B would be white. So I'm going to have 3 black, 1 white. Okay, so this little guy is going to be the only white one. And it's really important to kind of pay attention to your ratios. These are going to be very helpful as you move on and kind of go through, because when you see certain ratios, even if they're not perfect, so say this is my cross and I happen to get, let's say I had 7 black, okay, and I had 3 white. Okay, this is pretty close to a 3-to-1 ratio. It's not perfect. It's never going to be perfect, but it's going to be pretty close to a 3-to-1 ratio. So that way you can infer, okay, if you see this ratio, that the parents were probably heterozygous. Okay, sometimes you have to work backwards, not only forwards. Okay, so this is Mendelian inheritance, simple dominance with 1 trait. In the next video, I'm going to talk about with 2 traits. So let me know if you have any questions.